intelligence. Okay. So here I'm going to list just a few nuances in the context of visual processing where the humans and the current machines are doing fundamentally different computations for a similar task. And in these examples, I would like to emphasize the role of perception, to highlight the role of perception as a human capability. And I would like to advocate the perception as a new dimension or further dimension that we want to use to set the standard for the performance or capabilities of artificial intelligence systems. Okay. Here is the first example. The state-of-the-art AI has no problem with recognizing this as an image of a lion. But once the lion is put in a low contrast scene where the brightness features are not salient anymore, the same algorithm may fail. But the human visual system can adapt to the new scene statistics in a few tens of milliseconds. And it is to create a perception of the visual scene where the information of survival value, like this dangerous animal, can be immediately salient. Here is another example. It is a very easy task for an image processing system to recognize white and gray colors in this scene just by measuring the light emitted from these surfaces, for example. But this is not always the way that we as humans see colors. Okay? Here is an example of a color detection task. I'm going to ask you that tell me what the color of the top and the bottom surfaces in this image. Okay. It is easy, right? Top is gray, bottom is white, all agree. Now I cover the join area of these surfaces here by just a rectangle. Okay, both surfaces are gray, because we are doomed. How come we fail to detect the actual colors? Indeed, our visual perception took into account the context information in this example and create this illusion of color for us. Is it considered a failure or it is a useful illusion? This is a matter of investigation, right? Another example. The state-of-the-art object recognition algorithms can, to a large extent, detect real-world objects independent of their orientation, size, or viewpoints, similar to the example images of a car in here. This capability has been enabled by virtue of enormous amount of training data fed to these artificial object recognition systems, and they can be trained on differentiating between objects or detecting objects, no matter how their physical properties has changed, okay? Like this orientation, size, or viewpoints. But this is not certainly how humans learn invariance in recognition or recognizing arbitrary real-world objects. It's not just by enormous amount of training data that we need to be exposed to be able to, for any objects, to be able to say, no matter what the viewpoint or the subject or our distance from that subject is, if we can tell this is a car or something else. And this implementing invariance is an indispensable task of our visual perception. Okay, that's our daily life. This is how we look around. So we have to constantly implement invariance when we are perceiving the environment, the visual environment. Okay. Now let's revisit the example of muffin and chihuahua. while an AI system can be challenged by these very similar images of this type of dog and the muffin, our perception works fine without even being aware of this complexity. It's a very effortless job for us to differentiate between these two objects in all these images. And lastly, getting to the cutting edge AI application, autonomous vehicles. The main challenge here is how an intelligent agent can navigate and process information from multiple sensors like lidars, cameras, GPS, ultrasonic sensors, dynamically and in a real world environment and in real time. Okay? For us, it is solely our visual system taking care of this complex task. And indeed, such capability involves an active representation and readout of the visual scene over time. That's how we can analyze and perceive the world around us and interact with the environment, like driving the car. As an exemplar of such active visual perception capability, is how our brain constructs the visual world for us during constant eye movement. So this is an image, and this is the trajectory of the movement of the eyes of and subject. And so this is how we interact with the world dynamically, and this is how actively our visual perception construct the visual world for us, similar to this autonomous vehicle example that is going to work dynamically in the given environment. So we have seen the adaptation as one of our perception capabilities. We have seen inference about color. We have seen invariance features of our perception, the complexity of the features that we can figure out, and also the dynamicity that we can handle in interacting with real world. And following on the topic of this active visual perception, the very last example around the idea of dynamicity or the ability of dynamic processing. In this talk today, I will be present a research in our lab which aims to decipher the brain algorithms by which we perceive the visual world robustly during our rapid eye movements, which can be also insightful for artificial systems that are operating in a dynamic scene. So let's start with the findings of our research with some introduction. Vision is a constructive process, meaning that our brain constructs or reconstructs the visual scene for us. As a matter of fact, this is one of the fascinating examples of this active construction or reconstruction. Even though we perceive the world as a stable and continuous, the visual input to the brain actually consists of a discontinuous slideshow of retinal images during our free viewing of the visual scene. This phenomena or whatever function is called visual stability or perceptual stability, and it is constantly happens across our rapid eye movements, also called saccadic eye movements or simply a saccade. And today's talk is about the efforts in our lab toward understanding how this constructive process is happening in the brain. What is the computations, underlying computations of the brain to make that possible? So our research is also, this presentation of this project specifically may be used to also demonstrate an example of a multidisciplinary research, which involves developing, extending, and employing tools and methods from machine learning, which I assume to be in line with the broad interest of this workshop as well. Okay, with that, let's get back to the research that is going to study this visual stability across saccadic eye movements. Let's first introduce some terms and concepts that you may not be familiar with if you're not in the community of neuroscience. So this, I am just explaining what the visual neurons are doing as we are watching the scene, as we are seeing the world around us. During fixation, when we are fixating our gaze to a specific location, when we are just fixating our eyes, our gaze somewhere, each neuron in the visual part of the brain, which is also called visual cortex, can see only a small part of the visual scene. And together, all the neurons in the visual cortex can tile the entire visual scene. But one neuron can have access to information from a specific part of the scene, a small part of the scene. And that area is called the receptive field of the neuron. And as the gaze moves, the receptive field of the neuron also will move following the gaze location. So this is the receptive field of one neuron in visual cortex, for example, when our gaze is in here. And now I'm just playing how this or what this receptive field area experiences during our free viewing or saccadic eye movements. Okay. So this is what happens during eye movement. As we move our eyes, the image that falls inside the receptive field of individual neurons. Okay, the image that, if you follow this white circle, is the image that is projected inside the receptive field of a neuron. It rapidly and drastically changes across eye movement. But as we all know, our natural vision does not perceive these huge shifts and jump as we move our eyes that actually coming to the brain or coming to our visual system, but we cannot perceive it. And instead, we see the world as a continuous and a stable scene during eye movement. Okay. So this is the real world example. Indeed, we move our eyes 3 times a second on average to scan the visual world around us, or to deploy our visual attention to regions or objects of interest all around us. And every time we move our eyes, these huge image shifts are projected onto the retina. The retina is a layer in the back of the eye, and basically what is getting into the brain is just these video frames that are captured by this moving camera. This is exactly the image that is coming into our brain. But again, we do not perceive the world like this. Otherwise, we have been we have been overwhelmed by the amount of the visual information, and also these not pleasant type of experience of visual scene during our eye movements. Okay. So what does the brain do to keep our visual perception of the scene stable during eye movement? First of all, the first strategy that brain adopts is that the retina, again, which is the very first layer in the back of the eye, that is the first layer of visual processing in our nervous system, the retina shuts down during eye movements. Okay? And this way, the image motion that is projected on the retina will not be transmitted to the brain. Okay? So this is just the ballpark of the times that the retina is shut down, which makes of effectively blind during eye movement. So considering 3 times a second that we move our eyes on average over 24 hours, it means that we are almost 40 minutes blind during the day. Okay. So this is the first strategy. Motion smear, jumps, shifts are not transmitted into the brain. But as a result of this blindness, and we should experience the episodes of blank images coming into our brain, being associated with these blind episodes that I'm talking about. So, but again, our perception is not like a slideshow of images interleaved by blank images. So somewhere higher up in the brain should fill in the gap that is induced by the blindness or the shutdown episodes of the retina. Again, it is just to be very brief and simple, the way that the visual information is processed in the brain is that just the visual information is projected, or visual scene is projected onto the retina, and the information in the form of electrical activity is transmitted into the brain, the thalamus and primary visual cortex, and from there to other areas of the brain. So if the retina shuts down, the information that gets to the brain is disrupted for sure. And because our natural perception is not disrupted, so we are expecting the rest of the brain, or somewhere in the brain, in the visual areas of the brain, to take care of filling that gap. Okay, so this is the hypothesis with some evidence that how the brain may implement this filling the gap. And it is proposed, and again, there are some evidence that the brain does that using a copy of a motor command. And a motor here is just eye movement signal, the movement of the eye itself, that is sent to the visual cortex. So basically, there are areas in the brain, like LIP, supracolliculus, and frontal eye field, that are responsible for processing eye movement. During eye movement, or during planning the eye movement, these areas will be talking to visual areas, and that will help the visual system to represent the visual information in accordance with the position or the location of the gaze, to provide that continuous perception for us. Okay, but what is the underlying computations that makes this signaling, this communication, this information processing happens in the brain? So the question is, what are the computations? What is the brain algorithm in implementing, when implementing all of these signal transmission and information processing? First of all, we need to go into the brain. We have to collect information or collect data in the form of brain signals during eye movement, and also visual and the visual sensory processing, to be able to see what is going on inside the brain. But the very first challenges that we have for studying eye movement or visual processing during eye movement is the short time scale of the phenomenon that is happening during the saccade. Each saccade, or eye movement, a rapid eye movement, is lasting only maybe 30 to 40 milliseconds. So you have only 30 to 40 milliseconds to collect the data that is informative about all the computations that is happening to make that continuous perception for us. Okay, the data is limited. Relative to the complexity of the things that is happening in the brain. And the other challenge is that it is also not very much feasible to measure perception and quantify perception of visual stability. So how you want to define stability, how you want to measure it, how you can train a human or an animal to report that if they see a disrupted or continuous scene. So together, although the data that will be coming from the experimental approaches will be very, very valuable for this study, these experimental approaches alone is not sufficient. So in our lab, we are combining the data from experiments with computational approach to be able to decipher the computations underlying the visual processing during eye movement. What is the general idea here? So first, we want to provide a quantitative description of the input-output relationship, when the input is just the visual sensory and the output is the signals of the brain. And that way we can figure out the computations, or understand the computations that is going on from or between the visual input and the brain signals. So what is the map? What is the quantitative description of the input-output relationship? So once we have this descriptive, we call it model of the brain, of these computations, then we use the decoding aspect of the brain, decoding aspect of the model to infer the perceptual effects. So basically, once you have the model, you can ask the model, given some brain signals, what the model think the input to the brain was. And that answer is going to be the perceived input, not the actual input, that disrupted input to the brain. And using this closed loop approach, the encoding and decoding combined, we can both understand the computations underlying the representation of information in the brain using this modeling approach, and also the algorithms for readout or extract information from this representation using these decoding approaches. So in the rest of the talk, I will go to more technical details that may be of interest of some of you, but I have to always first from the data, and data comes from experiment. So let's start with how we collect data first and what we mean by brain data. This is how we go into the brain and measure brain signals. This is a 16-channel linear array electrode. So with 16 metal contacts or channels that can pick up the neuron signals close by. So if a neuron or a bunch of neurons is close to each of these contacts, the signal will be picked up by these electrodes or these channels. What's called the electrode is going to be inserted inside the visual cortex, or part of the visual cortex area before and middle temporal area MT. So these are the middle area or middle cortical area of the brain in the hierarchy of visual processing, starting from the retina to V1 all the way to infratemporal cortex, and these are the middle area. V4 area, again, to be brief for those of you who may be interested to know why these areas have names and why they're distinctive. V4, generally speaking, is in the area that is processing the shape of the objects or the physical properties of the objects, like color as well. MT area is mostly processing motion information. V4 is along the ventral pathway, MT is along dorsal pathway. So these are different visual pathways that processing what and where information, again, generally speaking, in the brain, and we are recording from these areas. So what they mean by recording. So let's see one of the example recording that we do in the lab. So here you see a visual stimulus, which is this moving bar. This is a Gabor type of moving bar. And what you hear is the response of a neuron. So basically you are listening to a neuron. Here, it is the electrical activity that is picked up, let's say, by one of these channels. So it is the response of a single neuron. And if you connect that amplifier that basically amplifies this electrical voltage to a speaker, that is the sound that you hear. So basically you are listening to a neuron. If I just want to explain what we see on the screen, this is the snapshot of one of the, it is a snapshot of the recording that is appear on the oscilloscope here. So here is the recorded voltage. You will see these pulses in this image. That is these are the signals that the neurons use to communicate with other neurons. And you see the time scale of being 1 millisecond. So these are very, very fast events. If I just want to zoom in on one of these signals, this is the extended time scale of this signal. These pulses are called action potentials, and this is a waveform on one of the action potential. Again, this is a zoomed in version, so the time scale is just one order of magnitude less even. How this recording will be used. So look, you could think of applying a threshold. If all what matters is the fluctuations, you can think of just applying a simple threshold, even a constant threshold, and detect the time of these high fluctuating voltage events. And you just mark those times. So if you go maybe by these alignments, so this vertical bar is corresponding to this action potential, this one is going here, this one is going here, so on and so forth. So basically this way, all these electric continuous or analog voltage that is generated by the neuron, when it is communicating with another neuron, it is just the time stamps of, or reporting the time instance of these action potentials. And indeed, when we are going to use this data for further modeling or computational studies, this will be translated into a time series, a binary time series of zeros and ones. So basically, this is the language of the brain. So these zeros and ones, so all these scenes that are projected onto the projected onto the retina, going into the brain and creates our perception, is the result of these zeros and ones sequences and that are generated by the neurons throughout the brain. Next, I will explain what was the visual input to the brain and what was the eye movement task that we use to collect this data, to record activity in the brain. First of all, we are using the macaque monkeys for this study. And those recordings that I mentioned from area before and MT are done in macaque monkeys and non-human primates. And basically, the advantage of having monkeys for these studies, the monkeys' visual system is very similar to the organization of the human visual system. So if there will be understanding in non-human primates, it will be guiding how human is also processing the visual information. And by the way, because all these recordings are considered invasive methods, it is not feasible or even ethical to be done in humans. So monkey is what we have, and the data that's coming from monkeys will be very valuable. So here I will just explain the task. What is going into the brain? So this is the screen of a monitor that is in front of a monkey. And the monkey has been trained to first fixate on the central fixation points. So this is basically showing the gaze, the location of the gaze of the monkeys, which is a central dot on the monitor. And then This is the start of the task. The monkey fixates for about 600 to 700 or 750 milliseconds in here, and then the task starts. So these visual probes are presented during fixation. The fixation point turns off, and that is the cue for monkey to move his gaze to the second fixation point, which is here. And it fixates here for another maybe 600 to 900 milliseconds. And this will be considered successful trials, and the monkey will be awarded by a drop of juice at the end of the trial. So I will play this again. So that was the start. So these probes, these white squares are presented on the screen. Fixation point turn off. Monkey move the gaze to the second fixation point. These probes continues to flashing. Monkey fixates on the second fixation points for a while, and it will be rewarded by the drop of juice. So that is the eye movement task plus visual stimulation. So I would like to emphasize that these white probes that are presented in random location at random times. So basically, if you are familiar with the system identification in signal processing or in machine learning, this signal, this visual input is considered a spatiotemporal white noise. And it is very important to have the response to the white noise kind of a stimulus to be able to have a model of the system that is not biased toward the properties of the input, because now the properties are randomized, there is no specific patterns, and basically white noise is going to provide, in theory, infinite number of spatiotemporal patterns, and that we will be probing the brain, and we can record the signals when those many patterns are presented to the animal or to the visual system. And another thing to note that, so this is the slow motion kind of version of the presentation, and the probes presentation is just 7 milliseconds. It is at the refresh rate of the monitor, so it is very brief probe presentation, very fast probe presentation that will be appearing on the screen. So lots of spatiotemporal patterns will be generated in this way. So next, now that we have the animal trained, we put the electrode inside the brain while the animals is doing the task, and this is the recording. So basically, each contact could pick up a neuron, and usually we can isolate around 3 to 4, 5-ish single neurons from each session of the recording. And here, so basically each of these blocks is associated with recording for one neuron. So neuron 1, 2, up to neuron 5. And multiple lines for each of these neurons is representing multiple trials that the monkey performs during an experimental session. And roughly the number of trials are in the order of hundreds, a few hundreds, and even thousand. So we have multiple run of the same task over and over, and we are recording simultaneously from multiple neurons at the resolution of sub-millisecond from the neural signals. So the recording using these electrodes, and the sampling rate is 40 kilohertz, so it is on the order of 0.1 millisecond resolution that we collect this data. This is exactly those 0-1 pattern that I mentioned in the previous slide. So basically these are ones and zeros for each trial of the experiment, and you have a bunch of these ones and zeros, or we call it the spike trains, collected for individual neurons. So these are, again, that's trials, neurons, and these are 5 example MT neurons, and the time again on the order of millisecond. We also trace the position of eye using the eye tracking system. Again, it's a very high resolution system, 2 kilohertz sampling rate, meaning that every 0.5 millisecond, we know where the eyes are or where the location of the gaze of the monkey is. So we have these eye position data, we have these recording from the brain, we have control on the presentation of the visual stimulus, those white squares on the screen, where the cue is presented, where fixation is happening. So with all these precise measurements, with high temporal resolution, we hope to be able to find out how brain translate the visual information and motor information to these zeros and ones signals. Okay. So this is the actual task of the quantitative approach or the computational approach. So we have those experimental data, again, high temporal precision. Now the question is that what is going on in between in the brain from the input, which is motor and visual, and the spike trains. So answering this question is basically by developing encoding models, that I call it here. So if you have a general input to the system and general output to this out of the system, then what is the map in between? What is the system that does this transformation? That's what we call encoding models here. You could think of different ways to figure out this map. In our lab, we are doing statistical approaches for all of our modeling. I'm a statistician, so my interest is to have the statistical probabilistic approach for deciphering a system that is actually stochastic, is actually random, or we have no choice but to model it as a random process. So this is one point that I want to emphasize here, again, for those of you that may not be familiar with the neural signaling. And these zeros and ones are basically stochastic signals or random signals because of the noise in the system. So brains, the biological system, there are lots of chemical and mechanical activities going on that will be influencing the generation of the spike patterns. On top of that, which is even more important than this kind of uncontrollable noise, is other factors that are not measurable or even not observable to the experimental that is affecting this generation of spike trains. For example, the mood of the monkey. Is it tired? It is active? Or if the monkey is attending somewhere, so listening to somewhere. These are not the input to the model that we are trying to figure out. The model is just taking the visual input and the eye position. We do not have control, or we cannot measure other factors that are simultaneously influencing the spike train. So we have no choice but considering or treating this system as a stochastic system. So that was the statistical approaches or probabilistic approaches are very much making sense in studying the neural signals. So encoding model is going to figure out what transformation can reproduce or capture the likelihood of data given the input to the system. Okay, so for those of you familiar with these statistical terms or notations, that is the likelihood of the observation or the output. Now let's look at what we are facing when we are trying to figure out this model. I talked about the stochasticity of the data and system. I talked about the discreteness of the data, these binary sequences, which can challenge any modeling efforts. Even the state-of-the-art statistical learning methods. We are talking about high-dimensional data and high-dimensional systems. So you could think of, for example, the dimensionality of the input. If you can just have a normal 1,000 by 1,000 pixel image, which is varying over time. So you have 1 million pixels over time with a resolution of milliseconds. So you can do the math and see how many visual dimensions that you will have. So you have that eye position, again, every half a millisecond that you want to also incorporate in your input. The dimensionality of the output is also high. So you have, again, millisecond resolution data across multiple neurons and multiple trials. So hundreds of trials, tens of neurons. And even for 1 second of the experiment, only 1 second, you have thousands time points of these zeros and ones. So you could think of high dimensionality will challenge developing this encoding model. Discreteness of the data, stochasticity of the data and system will also challenge the modeling. And the goal here is to have a model that transforms this high dimensional space of input output to a low dimensional feature space in order for us to be able to understand and interpret whatever is captured here. So we cannot work this high dimensional space, as many of you in the field of machine learning are familiar with. So we need this low dimensional feature space. That is what we call model. And what other things that is specific to the brain computation is that this processing, this transformation is nonlinear. This is, again, another challenge for any modeling and any model development. Now getting more specific to what we do. So here, and this statistical framework that we use for encoding model, we start with the classical generalized linear model. In the generalized linear model, so you have a one linear processing stage and one nonlinear processing stage. Okay, so you could think of this linear processing and a bunch of filters, and the nonlinear, which are transforming the output of the linear part to the actual response of a neuron. So this nonlinearity will generate the rates of these zeros and ones, or basically rates of ones generation, and then using random process generator, in our case, which is mostly Poisson spark generator, we can go from the rate to the actual spiked rates. Now with that, this is the general framework. I want to emphasize that this, for the specific task of visual computation during eye movement, this is not sufficient. And indeed, basically, I forgot this part, that this modeling is nothing but having this probability formulated as a function of input and the parameters, which are comprising the linear components and the nonlinear components, and maybe some general offset or some feedback components. So given these parameters and the input, the function that generates this likelihood of the output or the observation will be our encoding model. So I was going to start with the idea or that this model for the specific task of visual processing during eye movement may not be sufficient. And why? Because movement is a behavior. So we are talking about the interaction of behavior and sensory, generating the brain signals. So once you have this, it means that the brain processing is actually is a function of the state of the brain. The processing is a function of the state of the brain. So the model and this transformation will be varying in time because of this changing states. And so do the model components, the linear parts, the nonlinearity parts, this part and this one. Okay? So we need a modeling framework that can capture the time varying or the non-stationary properties of the system. And actually, we want that time varying capability to be on the millisecond resolution, or a few millisecond resolution. Again, eye movements happen only as short as 30 to 40 milliseconds. So all these mapping should be figured out using a data that is collected in that short time window. So now we are even adding more to the complexity of the model. So that time varying is one thing. Because of time varying, the dimensionality of the model will be even more increased, even in an infeasible extent, or that we are adding dimensionality to the system, because now time is coming into play. So at each point, with respect to the onset of eye movement, you will have a different system. You will have a different model, because it is going to vary by time. So all these parameters that I introduced here, because they're also varying by time on a millisecond resolution, and the number of parameters will be high, and comparatively, the amount of the data that is available to figure out those parameters is limited, and we are working in the sparse space of data because of the high dimensionality of the model. And on top of that, because now the model is very high dimensional with a very large number of parameters, we have to be taking care of the generalizability feature of the model. So you could think of having an underfitted or overfitted model because of this discrepancy between the amount of data and the number of parameters, and we have to always take care of that, because if you remember, I told at the very beginning, our approach is to have the modeling encoding model first, and then the next is to use the model to decode information. So for that decoding, we have the model needs to be generalized to unseen data. So that is an important feature that we have to take into account. With that, I will just take you through the results. So using the data that we have been collecting, and also using the modeling framework that I just introduced. I mean, it is just an extension of the generalized linear model to accommodate non-stationarity of the system. And I would like here to just show you how we are going to understand the computations underlying the visual processing during eye movement. All these experiments or computations are done by a talented PhD student in our lab, Amir, who is now graduated and is spending a postdoc for a while with us. And all this research has been possible using the real intelligence and effort of Amir. So here I'm just summarizing the data part. So this is the experimental paradigm fixation and saccade, making saccade, and the second fixation and the recording areas. Here I'm showing a time axis where 0 is referring to the onset of the eye movement. Okay, so 0 means the onset of the saccade. And here it's after, times after, and here is times before. So now, and we are collecting a spike trains, like these are all zeros and ones that are collected for one trial across time to saccade from one neuron. Okay, now this is the actual part to start figuring out what we do to be able to figure out the computations underlying this input and output transformation. So let's have a quick tour. in the world of modeling. So the goal is to assess how neurons in visual areas, these areas, are representing visual words. Okay? For that, during eye movement, for sure. For that, we needed to trace the dynamics of the sensitivity of the neurons, or the processing of the neurons as it changes during saccade, okay, or during the eye movement task, from fixation, perisaccadic windows, and the second fixation. The neuron sensitivity, as I was highlighting, important to be traced dynamically across time to saccade, is defined as following. So at each point in time, it's actual time with respect to the onset of the saccade, and the sensitivity of a neuron is defined as the efficacy of a stimulus at certain location, present at certain location, and at certain delay with respect to this time, it's a specific time. So if a probe, it is presented a specific location at some delay with respect to this current time, so basically that delay is going in the reverse direction of this axis, how effectively it will be evoke a response in a neuron. Okay, so at the end, once you have that measure of sensitivity for all locations, all delays, and all time to saccades, then you can trace the dynamics of the neurons computation across eye movement. Now let's start with figuring out how we can compute that spatiotemporal sensitivity map. For that, we first discretize the actual spatial space and the temporal dimension. So the spatial, the location information, or the location dimension, and the time or the delay dimension. So this is how we discretize. We discretize the location into bins of 3 to 6 degrees of visual angle, and across time we bin the time into windows of 7 millisecond. Both this discretization, these bins of 3 to 6 dVA, are the resolution of these probes, these white squares, and this 7 millisecond bin is basically the resolution of the presentation of the probe, how fast they are being presented, which is every 7 milliseconds, again, refresh rate of the monitor. So first we do this discretization. That is the first step that can help us reduce the dimensionality, just by throwing away the data that is not at the resolution of the actual task. So this is simple, and that's the starting point. But if we do, even with this discretization, to get the full map, we need to assess or evaluate the contribution of 10 to the power of 7 of these units. I call it the spatiotemporal units, which represent location and delay for each time, to be able to evaluate the entire map. So 10 to the power of 7 is huge with respect to the number of spikes that we can collect in 30 milliseconds across trials and across neurons. So here we develop a dimensionality reduction algorithm using a statistical hypothesis testing approach that I'm not going to go into the details, and I'd be happy to elaborate on this during Q&A session if there was a question. But using that dimensionality reduction approach, we could select the STUs, or these spatiotemporal units, that are significantly contributing to the input-output relationship. And with that approach, we could basically remove more than 99% of these STUs, and basically we will be we remained with 10 to the power of 4 number of these STUs, and to be able to evaluate the sensitivity map across different time points. And we have been, it was very important for us for this dimensionality reduction to be kind of unsupervised, and to be able to, in an unbiased way, without any information imposed by us, to select the informative STUs. So that was the unsupervised type of dimensionality reduction using the statistical hypothesis testing that enabled us to start our modeling by reducing the dimensionality of the model. And here is the model. Here is the actual model that is going to be used to be able to fit or estimate that sensitivity map. So we have a low dimensional representation of the sensitivity map. They are together comprising the linear part of the model. So after fitting the model, we can figure out the weights of the STUs. These maps are showing the weights of STUs. We can evaluate the weights of the STUs across time to saccade, across delay, and for individual probe locations. So we have a spatiotemporal map that is varying over time, and that is the very significant contribution of this modeling approach that can, for the first time, enable us to model the spatiotemporal filters or kernels of a neuron across time with that temporal resolution. So the input will be passing through this linear part or linear filters, linear kernel or spatiotemporal sensitivity maps. And it is just a convolution operation, right? Just a linear operation. And then the outputs will be summed together across a space or a location. And there will be another kernel, and that is going to be capturing the overall activity of the network of neurons aligned to the time of the eye movement, the start of the eye movement. This will be added here. And the output of the feedback signal, which is going to capture the history effect of the spike generation, is also going to be added to the output of the linear filters. And then everything together will pass through a nonlinearity, which generates the rates of spikes. And using these spike rates and assuming a Poisson, inhomogeneous Poisson process for our spike generation, we can recover the spike trains. And then, so the model has been fitted on the actual spike trains, and all of these nonlinear parameters and the parameters of the feedback signal, feedback kernel and the offset kernel and these linear filters have been, all these parameters have been fitted simultaneously using this input-output data, and this is the actual output of the model. And then, again, I'm going to skip the details, but the way that we have been training this model or fitting the parameters was through a maximum point process. That's important because we have discrete data, a point process maximum likelihood estimation, and that is going to basically fit the model in a way, or estimate the parameters in a way that the likelihood of the observed data is maximized. It's a very intuitive measure, and again, it is tailored for discrete signals, and we could fit all these parameters robustly all together, and that is also important. So this is the outcome of the model. Okay, so here, this is exactly a few samples of these maps. They just rotate them, and this is time to saccade. This is the delay with, the time with respect to the current time relative to the onset of the saccade. And here it is showing the weight of STUs for the kernel or the sensitivity that is associated with one specific probe location. When it's a receptive field of the neuron during first fixation, this is the center of the receptive field of the neuron during second fixation, after monkey moves their eye to the second fixation. And for other locations, we can also, from the model, we can generate these maps. So here, basically, we can recover these three-dimensional spatiotemporal filters for each time to saccade, and these are the example times. So this before 0, it is fixation 1, after 0, it is fixation 2, and we see that the neurons is the temporal sensitivity of the neurons in response to something happening inside this receptive field 1 during fixation, inside this receptive field 2 during fixation 2. And we see that the neurons is sensitive to multiple locations. Here I'm showing 2 of them during around the time of the saccade. So this is basically the nature of the computations happening in the neuron that generates those zeros and ones signals. And we use it later for further analysis, but in this talk I just stop here and just do not talk more about how we use this information. Again, for those interested in modeling part of the work, we always, like other modelers, we have to test the accuracy of the model. Here is for sample neurons, sample spike trains, sample experimental session. And this is how data, how model predictions following data, gray trace is data, and the brown one is the model. So this is a spike train. A spike rate underlying the generation of a spike train, and we see that how well on the scale of single spike, single trial, single neuron, the model can capture the response of the system. So that was single trial examples across trials, across neurons, across sessions. We also evaluate the performance of the model in terms of the information per spike measured in information per spike measured in bits per second. And we show that no matter we are in the fixation window or the perisaccadic window, the model is performing well, although we know that the model is very much more complicated during saccade, but we have been avoiding all the overfitting or underfitting, depending on the questions, the modeling questions that you're asking. We have been avoiding all of these, and this is the performance on the unseen data, on the test data, and not used for training the model. We also and we show that no matter we are in the fixation window or the perisaccadic window, the model is performing well, although we know that the model is very much more complicated during saccade, but we have been avoiding all the overfitting or underfitting, depending on the questions, the modeling questions that you're asking. We have been avoiding all of these, and this is the performance on the unseen data, on the test data, and not used for training the model. We also looked at the prediction of the model on the response of the neuron to a specific probe location. Again, basically I'm avoiding explaining this plausible. At least the point here is that the model, which is dashed line, is following the data, which is solid line, no matter the response is generated during the response of the system. So that was single trial examples across trials, across how model predictions following data, gray trace is data, and the brown one is the model. So this is a spike train, a spike rate underlying the generation of a spike train, and we see that how well on the scale of single spike, single trial, single neuron, the model can capture the response of the system. So that was single trial examples across trials, across neurons, across sessions. We also evaluate the performance of the model in terms of the information per spike measured in information per spike measured in bits per second. And we show that no matter we are in the fixation window or the perisaccadic window, the model is performing well, although we know that the model is very much more complicated during saccade, but we have been avoiding all the overfitting or underfitting, depending on the questions, the modeling questions that you're asking. We have been avoiding all of these, and this is the performance on the unseen data, on the test data, and not used for training the model. We also looked at the prediction of the model on the response of the neuron to a specific probe location. Again, basically I'm avoiding explaining this plots, but at least the point here is that the model, which is dashed line, is following the data, which is solid line, no matter the response is generated during saccade. This is this colored traces, or if the response is generated during fixation, which are these black traces. So now we have a model and that has been trained on the actual spiking train. It is performing well on the unseen data, and it is providing us a generalizable, a predictive framework to go to the next step, which is the decoding part. Okay. So let's see what that is. So far, we have been fitting a model that could capture the likelihood of data, probability of y given x, the output given input. Now, we ask the reverse question. Given the output, we ask the model what you think your input is, and the answer would be the perception, the perceived stimulus. It is different from this input. And the goal and the hope actually is that once we know how this transformation happened in zeros and ones, then how we extract or infer visual perception information from spike trains will decipher the algorithm of the brain for reading out visual information during eye movement that is underlying our stable perception during eye movement. So this encoding is the brain computation. The decoding is the brain algorithm for generating our perception. Again, this is representing how you go from input to output, which is brain signal. Decoding is how to go from output or brain activity to what we call our perception. This is how this is the outcome of the brain. We don't know what is going inside the brain in real life. We only have access to the perceived stimulus. We don't even know what the actual input is. So whatever we see in our life from the visual, coming from the visual world, is basically our perception. The real world could be different, as that color illusion that I mentioned in one of my examples at the beginning of the talk. So we are reading out perception this way. And without explaining how we implement the decoding or the decoder or our decoding model, I'm just here showing the result of the decoder. So here I'm just trying to explain what individual components are in this demonstration. So this is the eye, and this is the location of the gaze. What is this dashed line? This is the first fixation point. This is the second fixation point. And this is that 9 by 9 grid across horizontal and vertical directions. This is presented on the screen, a monitor screen for sure. And then the z-axis is the latency of the processing with respect to current time. So this is the instant of time, the current time, or every instant of time with respect to the onset of the saccade. Negative means before, 0 is on the exact onset, and positive means after the onset of the eye movement. So at each point with respect to saccade, we can have a readout of visual scene, meaning that where information and how long and or with how long delay that information is processed by the neuron. So here we are well before the eye movement. The neurons is processing the whatever is coming inside its receptive field, which is this region, and that has been happened 60 milliseconds ago compared to this time. And now that I'm running the result of the decoding, we see that during fixation, so this is the times are going during fixation, and the information is coming from here. Very small information, or what's called, I would say very weak processing for the signals coming from this area of the scene, which is going to be next location of the receptive field when the eyes are going here. So this is one of the kind properties of the visual processing of the brain, to be predictive. Before eyes even start to move, the information from this part of the scene is starting to be processed, which is not at all trivial. It is one of the mystery of the computations that we are figuring out. So we are getting closer and closer to the onset of the saccade, and the eyes start to move. That's time 0. And then you will see the change of sensitivity. Again, here the second receptive field location is activated. Now it is more strongly. Eyes landed. We are 30 milliseconds after the start of the saccade. That's what I'm saying, that eye movements actually happening only in the order of 30 to 40 milliseconds. The delay by which the neuron is processing this part of the scene is now different and higher at 100 milliseconds. The neurons start to process the area around the saccade target, the fixation point 2, and that is with a different delay, around 140 and 50 milliseconds. And also it is still responding to something that is presented inside the original receptive field, although the eyes have moved, and the neurons should not have access to this part of the scene. So you will be seeing here multiple location, the sensitivity or processing information coming from multiple location at different delays at each point with respect to the onset of the saccade, and that is the way after onset of the saccade, and the neurons is only seeing inside its receptive field too. So this time precision in reading out the location information during eye movement is the first finding that can enable us to be able to figure out how the brain is making an integrated or a stable visual scene by this way of representation and reading out information about the visual scene, which is not at all trivial. And again, for the interest of time, I'm not going to basically explain the algorithms that we understand from this processing, this dynamic processing. That is going to be the brain's algorithm of our perisaccadic perception. Okay, so to recap, I was talking about the encoding, the combined encoding and decoding approach, or modeling approach that we have been using here, combined with or integrated with the experimental data to be able to decipher the computations happening in the brain, and also reading out or inferring information from that computations. And this is what we understand, which is a rich descriptive stimulus-response relationship and computations underlying our visual perception. And the ongoing research in our lab is trying to extend and those readouts of single neurons to the population level, because we know that at the end of the day, our perception is coming from the activity of the network of neurons, and millions of neurons, hundreds, thousands of neurons that are contributing to our perception. So we want to know collectively in a neuronal network how this information is processed and how perception is read out. And not only it is, this approach could be used for visual stability during eye movement, we can also figure out other perceptual inference, for example, the time perception and the location perception. There are lots of interesting phenomena that is happening during our eye movement. For example, we think the objects are happening faster, so we have time compression, we have space compression, we have this illusion of objects are closer to each other and than what they are actually, what their actual physical distance is. We are mislocalizing objects during eye movements. We cannot, depending on at what time with respect to the eye movement we are and where that object is, we may not be able to report the actual location, the correct location of the objects. So all these approaches could basically tell us how we perceive time, how we perceive location in our natural vision. And natural vision is our dynamic vision when we are interacting with the visual world as we move our eyes. Okay. And the ultimate goal of the lab is also, aside from this basic neuroscience and also algorithmic level of the visual information processing, we are going to transfer this knowledge to enhance artificial intelligence or artificial systems that are performing similar visual tasks, okay? And that are capable of implementing our perceptual abilities rather than just our sensory processing. And we are also aspire to design the next generation of brain-machine interface or biomedical devices like neural prostheses that are able to restore or reproduce our natural vision outside of the brain using control devices or by interfacing with the brain, for example, in prosthetic applications. And with that, we are trying to translate the knowledge from brain science to the actual engineering or computational science. And this is a general pipeline of research if you are going to pursue artificial intelligence related research or machine learning related research in the computational neuroscience lab. So the start of everything is the data, and then we have data analysis. So it's the data science part of the whole pipeline, and then we have computational modeling in terms of encoding and decoding. So the inference part as well. And once we have go through this pipeline of it's called the individual elements of the research, we are trying to basically answer or decipher the principles of the brain computations. And particularly our lab, or maybe other modeling labs in computational neuroscience, trying to advance methods from signal processing, computational modeling, and machine learning, also statistical inference and estimation methods, to be able to get the most meaningful, generalizable, and accurate model of the brain at the level of both encoding and decoding. And for those of you who are interested in this field, this is a very great time for neural data science or computational neuroscience or whatever the brain computation studies. Because of the massive data sets available, because of the advancement of neurotechnology. So this is the type of data that we are using in our research, multi-electrode array. There are lots of other brain measurements, tools, modalities, and devices that can generate enormous amount of data. Now we also have very fast and powerful computers that enables us to process this information. But this is our main role. This is our major responsibility, to be able to make sense of this data in order to figure out the principles of the brain. We have to handle the high dimensionality, stochasticity, and large scale, non-stationarity, discreteness, sparseness, and nonlinearity of either data or the system. I hope that you have been, at least some of you, got interested to what actually the brain algorithms are to implement the human-like intelligence. And with that, I would like to just revisit the concept of human-like or less artificial intelligence that I brought up at the beginning of this talk. Again, by presenting the findings of our research about the brain computations underlying visual processing across eye movements, I try to incentivize the idea or emphasize basically the importance of studying the basic science of intelligence alongside the advancing the engineering of intelligence. So what intelligence is, this is the first thing, and then how we can engineer intelligence, and for our case specifically, how we reverse engineer brain to make those artificial intelligence or outside the brain. So this is another example, my last example, to challenge the artificial intelligence in terms of possibly ignoring the natural, human-like intelligence. So we know that lots of interesting applications, including visual processing or vision applications, that are enabled by the advancement in AI and in machine learning. But if you notice, most of them are enabled by increased computing power and also the availability of large data sets. And with, to be honest, a small number of incremental improvements in the technical or methodological aspects of machine learning. These are great achievements for sure, but I'm going to emphasize that these are certainly not sufficient. And this is how I want to highlight the complementary role of basic science intelligence and engineering of intelligence. This is a recent breakthrough in AI. Many of you may have heard of this AlphaGo competition by Google versus the world lead, whatever, a player or a top player of the Go game, which is a Chinese game, by the way, Neda, and that in which in that competition that supercomputer or AlphaGo Google software beat the beat Neda. I think it was in the series it was 4 games to 1, and that was again a breakthrough. Now see on the other side of the coin that I'm going to basically bring up here. Okay, all these intelligence, all these powerful computations happening on these sides, on the AlphaGo algorithm or software, is in computers. Individual computational elements of these computers runs at clock speed of millions of cycles per second. And the computations are via electrons. Electrical signals are transmitted at the speed of light. Here we have a brain that works with zeros and ones, those spike trains. And the speed of the propagation of the spikes is less than 1 meter per second. And at most it is 100 meter per second, depending on where the brain we're talking about. And so basically with this speed, the visual information, even it takes maybe 200 milliseconds to get from the retina, which is the first basically image sensor, the first layer, and it's an image sensor, to get to the higher areas in the brain. So by the standards of a silicon computer, such propagation time are glacially slow. And basically you could think of around 10 million times slower than these computers. So brain technology as a computing machine, the hardware of the brain as a computing machine is a very low technology, or at least a low technology comparative to this kind of technology. So again, as I said, the computer's processing is via electrons, and the brain's processing is via ions. And basically it takes a huge flux of ions for neurons to generate spikes. And ions are much heavier, I think around 70,000 more massive than electrons. They're extremely larger and extremely slower than electrons, yet our biological vision works in real time, okay? Second, the biological systems are noisy, including our brain. There are lots of chemical and mechanical activities and movements happening in the brain. So it's a noisy signal that we use to process in the world around us. This is why the electrons, the CMOS computers, the digital computers, are so precise, and with a bit of noise they will crash in terms of, or fail the implementation of the task or the application. Yet our biological vision works in a very robust way. So here I just wanted to contrast the two types of intelligence. For sure, our brain and artificial brain are going to, or artificial intelligence are developed for different purposes. It is not always that we want to go to the human intelligence, because also we know that there are cases that similar to this competition, that the supercomputers or artificial intelligence beat human. But I just want to trigger or highlight the idea that what is making us powerful, what is making us to have to construct the world around us, to generate perception of the world, is the brain algorithm, is the brain software. So we are, although the most power for the current AI is coming from large data sets and super fast computers, here we are talking about algorithms, okay? And it is, I mean, it's a very good time for computational people who are also interested in biological or basic science research to jump in and try to add a whole new dimension to how we define or even redefine or assess the performance of an artificial system by inspiration obtained from studying the brain and reverse engineering the brain. And I'm just my 2 cents here is that that would be very influential. Okay, well, that is just my opinion. So with that, I would like to thank everyone that made this research possible, especially Amir for all the works, experimental and computational he has done. He was also co-supervised by Dr. Nodust at the U, and then all the experimental parts have been enabled by collaboration with Berad's lab. And there are other lab members that have been also directly or indirectly contributed to this research. And we are very, for sure, thankful of the entire lab for the findings that I presented today could not be possible without their time and effort. And also the funding agency that has been contributing to the money side of our research. And thank you for your attention. And I look forward to seeing you in the Q&A session. Thank you.
Talk. It was very informative, at least for me with no prior knowledge to the area. It was very interesting. Thanks a lot. So now we have time for question and answer. So please type in your question here, and we will share it on the screen.
Start, because I think I have been getting some typed question, and I try to take a note of them. So maybe I can start answering those that I already got. Okay, so you want to put up the questions one by one? Sure. First of all, hi everyone. It's nice to meet you even virtually. Hope this whole pandemic will be gone as soon as possible so that we can just meet in person. So yeah, I will start answering the questions one by one. The first question is about if we are going to integrate visual perception and language processing, not in our lab. And that is not our expertise for sure. And in general, I think visual and language are working together, but I think individual of these research are so complicated that even their interaction, their interaction could be even possible at this stage. But yeah, for sure, there are researches going on, mostly at the psychophysical level, not the neural level, at the level of psychology information. But it is possible, but not by us. I'm not educated in that field, and maybe I don't have access to resources in that field. So they're asking, can we do brain science from a mathematical point of view? First of all, this talk was demonstrating one of the cases that how computational approaches could help experiments, to complement experiments, because you can always collect data, but the next step is to extract information from data. And that part is where quantitative approaches could be helpful. Yeah, graph modeling could be another framework for modeling, but just be careful that each question will take its own relevant modeling approach. So not all models are going to be helpful for all the questions that we are asking about the brain. Yeah, but we have graph modeling approaches in brain, mostly in the context of brain neurons connectivity. When we are talking about networks of neurons, how they are communicating with each other, there are lots of graph-based models that are used in the literature. And yeah, there are lots of mathematics involved when you are going to interpret data and model data. Okay, so they're asking that when the visual input is getting into the brain, is it in the form of continuous signal or a discrete signal? As I mentioned, the brain communication is in the form of voltages, electrical voltages that neurons generate. There are information aspects both in the analog part of the signal and also in the digitized part of the signal. Most of the oscillation studies in the brain are working on the subthreshold electrical voltages, which are continuous signals. In our study, we worked on the spike or action potential part of the signaling, and it is discrete. So it is not us that are discretizing the brain signal. The brain signal is discrete. And that is how it is challenging the modeling approaches, the models that you have been, working on in the literature of the athletics machine learning. Most of them, like regression-based models, classification, they are working with continuous data or labeled data. Here we are talking about the stochastic signals, which are also discrete, which are also sparse and coming from nonlinear system, and that is all complicating the whole process. Okay, this question is about the number of neurons that we need to record from to understand the brain computation. Yeah, I wish we could have access to large scale recording from the brain. That's one thing. The neurotechnology that is basically one of the trends now, developing advanced neurotechnology for accessing or measuring the brain is one of the active researches. But on top of that, it is not just the amount of data that we collect. Even for the single neuron level data, we still are struggling with how to interpret zeros and ones. So yes, for sure, multiple neurons are contributing. As I mentioned at the end of my talk, the network level analysis is also one of our future, actually ongoing direction. So we are expanding our models to population level and neuronal network level. It is possible, and we are doing, but even at the level of single neurons, that is a huge amount of information that still we are struggling to handle. Yeah, that was the hypothesis, and in the literature. And so there are two components to visual stability in terms of hypotheses. Again, there are evidence, works for that. But the first one, it is maybe a sequence of hypotheses. The first one is that the brain and input to the brain is suppressed or disrupted. So that's why we are kind of blind during eye movement. The next hypothesis is trying to stitch the pre-eye movement and post-eye movement scenes to be able to bridge the gap associated with blindness. Here, I was motivating the whole idea of visual stability as it is studied in the field so far. Our approach is going to be providing the computations from scratch. So we are not assuming blindness. We are not assuming filling the gap. We are basically deciphering the nature of the computation of the neuron. So in our model, in our data, we didn't assume any of these things. From the readout, we could understand that how those zeros and ones, which are the language of the brain, could basically inferring information about the integrity, about the stability. So basically, we are measuring stability, we are measuring integration using our decoders. Again, these are the hypotheses in the field, but our modeling approach, our data are not assuming this. But these findings or these hypotheses have been a guiding basically direction for many of the researchers, including ours. It is very insightful. No, actually, we don't want to sacrifice the resolution of the data just for the matter of dimensionality reduction for doing modeling. Model is not the goal. Model is just a tool to understand the brain computation. So we are not going to sacrifice data just because to develop a model. So we keep the resolution of data in milliseconds, which is the actual time scale of the eye movement and data and visual processing during eye movement. And the way that we try to reduce the dimensionality in the model space, not in the data space. Data is as is. And in the model space is to find some unsupervised or unbiased approach to get rid of the uninformative dimensions, those that uninformative about the that integrated perception or that readout of the visual scene. The uninformative about the relationship between the input to the brain, which is the visual information, and the spike trains, those zeros and ones. So the dimensionality reduction is part of the modeling, not affecting the resolution of the data. And it is just for us to be able to interpret the encoding model and also the decoding algorithm. Yeah, there are lots of works around bridging between brain science and artificial intelligence. Most of those works are published in neuroscience journals. And there is no conference in neuroscience, actual conference in neuroscience, so everything is journals. So if you look up for any deep learning or neural network approaches or machine learning, just search the database of neuroscience journal, Nature Neuroscience, Nature itself Science, what's called the J Neuroscience, all these venues, journals, that you can find the connection between brain science and AI in terms of the sample works of different research labs. In our case, I mean, we are also publishing in these kind of journals, not kind of classical ICML or CVPR, this kind of things. So if you're interested, you may want to look up it in the neuroscience journals. So there are lots of computational things covered there. It's not just biology. Okay, so the modeling approach that I was introducing here could be used for object detection and classification. And what I mean for here, we are also doing detection and classification during the decoding. So when I skip the details of the decoding. When I say we decoded visual information from using the model, it means that we presented some predicted spike trains to the model. And then we ask the model what you think the input was. And the input that we designed for the decoder was actually the detection task and classification task. So we ask the model, can you do detection? Can you do classification? With what performance? For what locations? At what specific time? And what latency? So all this happened during decoding. But it was for the purpose of this work, which is going to inform us about the location perception in that sense. Object detection in general, it is cat or dog. It may be, the other people are doing in computer science and machine learning. They are not working with the spikes. There is no zeros and ones. It is not mattering for them that what is in between. I mean, you have different layers, different parameters, different filters, different ReLUs, and they are making prediction about having something to be cat or dog, but you never know what is going on in between. You just have a bunch of layers and parameters without being able to connect it to the actual brain processing, because they are very kind of a black box most of the time, not always, most of the time. Deep learning, it is possible that you can use deep learning approaches. There are works, and again, the journals that I was mentioning, there are works that using deep learning for studying brain. Again, it depends on the question that we are asking. Not all the models, not the learning frameworks are appropriate for every questions. Deep learning is basically one of the popular frameworks, mostly in cognitive neuroscience, like dissection task, object recognition task. It is one of the favorite frameworks. Nonlinear model deep learning, yes, it is possible. In deep learning, you have ReLUs, you have those thresholding or whatever sigmoidal nonlinearities. So the system is inherently nonlinear, but this is not helping you if it is just it's a blind deep learning. I mean, if you do not have an informed deep learning or deep network, then the information coming from the model is not helpful. It's not toward understanding the brain. It's not toward understanding the actual computations, actual algorithms in the brain. Again, those models require tons of training data. Brain is not working with training data. Once we see the one instant of the object, we can figure out many aspects of that subject. So this idea of having lots of training data, having lots of parameters, it is not how brain works. So again, depending on the question that we are asking about the brain, this could be insightful, this deep neural network, but not for all the questions. The end layer of the decision for real world events. I'm not a control person, so I'm not maybe familiar with all the tools or approaches in fuzzy systems. So this is maybe not a good question for me to answer, but what we are mentioning here in terms of model, it could include any appropriate modeling framework, including maybe these fuzzy systems. Yeah, it is one of the system identification methods I'm not familiar with, and maybe I'm not a good person to answer this question. You're welcome. Okay, so in AI, they say that we usually try to reduce and separate perception and brain constructiveness to discrete signal and ignore the integration interdependence naturally existing. I am not sure if I understand the question correctly, but I mean, to my best guess, I think the question is about during in AI approaches, we are ignoring and the perception capability. That was basically one of the introduction slides, or maybe the start of the talk, to motivate the idea of human-like or brain-like intelligence, and by adding the dimension of perception. So again, the go-to standard for AI system could be the performance of human. Okay, although we have many AI systems that surpass the capability or the performance of the brain for some specific task. Yeah, what I was trying to convey in this presentation is that by adding the dimension of perception or any cognitive ability of the brain, so because my lab is doing visual processing, visual perception was the cognitive ability of the brain related to my own research. This is one of the cognitive abilities. So if we can add those kind of more natural processing of the brain or constructive processing of the brain, that could be more toward the human-like intelligence. It could be a drawback for AI if the goal is to be a human-like intelligence. But if the goal is to just perform a task perfectly, then why do you need to replicate the brain? Most of the time, you do not need to replicate the brain. But again, the examples that I was trying to mention at the beginning of my talk was toward the idea of that there are insufficiency in AI that cannot capture some of our capabilities, visual processing capabilities, because they are ignoring the constructive aspect of our visual processing, which is perception. So we are not just processing the word like a camera. We construct the word the brain construct the words for us, and perception is the field that is about that process. Yeah, there are works around that. The deep learning could mimic the computation of the brain. Again, it could be as naive as looking at the brain as a hierarchical processing. Yeah, visual processing in the brain is hierarchical. So it is going from retina to thalamus to V1, V2, V3, MT, V4, IT. So there is a hierarchy in the brain. So you could think of different layers of processing. This is physical things that is existing in the brain. And so neurons are having, I mean, if you want to model a neuron, or in reality even, a neuron is a linear processor and a nonlinear processor. And all the components of deep learning neural network is just linear and nonlinear components. In that sense, it is close enough to the structure of, not structure, to the individual elements or the organization of the brain, but it is not necessarily figuring out the algorithm level, the software of the brain. So I mean, having lots of LN, linear, nonlinear cascades and layers, it is how brain is organized, but the computation and information is way beyond the structure.
Talk, Neda, and question and answer. I also have some question, but I will send you an email. Thanks a lot for your presentation, and other participants. If you have more questions, please drop me a line with Neda, and hope to see you next time in Tehran, not virtually. Thanks a lot.
Yeah, thanks for you. I mean, listening to my talk, and I try to type my answers in the chat dialogue. I mean, if it is still possible after the meeting. I mean, if it is possible, I can start typing answers to unanswered, undiscussed questions. Do you want me to do that?
Well, I'm not, perhaps the technical team can answer that, but maybe we can do that. Or yeah.
Okay, yeah. I mean, everyone, just feel free to email me if you have questions. My first name, not my last name, @utah.edu. I mean, you can find me easily.
Okay, thanks a lot. Thanks.
Okay, good. Thank you. Thanks. Bye, everyone. Thanks.